My name's Kath Glidden, and today I'm here to talk about the 3-minute thesis competition and using it for attentive coursework. This was co-designed by me with excellent insight provided by Dr. Pugh. Sarah Ireland is a pharmacology intercalated student, and it's her work that we will be showcasing. So, 3-minute thesis, it's a competition for final year PhD students. What they have to do is communicate their thesis in 3 minutes to a general audience level, and that's the really important part, is to get the students to practice communicating to the general public. So the rules are just about to be shown. Yes, really simple, 1 slide, no animation, okay, 3 minutes max, and it has to be spoken. So why teach science communication for the general public? Why is that important? Well, it's hard, and there's lots of misconceptions out there. For example, only 1 in 7 Americans actually appreciate that the majority of climate scientists agree that's humans causing climate change. Another example is the misconception, or the alternative fact, that vaccines causes autism. Now, in a democracy in theory, it's the public's understanding of science, or lack thereof, that will alter science funding and government policy. So if we train the future scientists to be better communicators to the general public, fingers crossed, we should get better government policy, but also better communication with the public. So how do we do this? This is a paper published in 2016. They talked about using authentic coursework, hence the 3-minute thesis. They talked about using oral communication, hence the 3-minute thesis. I would include another principle, which is the art of storytelling. That is using an everyday object to explain scientific concepts. Like, for example, the mitochondria are always called batteries because the mitochondria provide energy to the cell. So what Kirsten and I did is we got the students to write a grant proposal in Alzheimer's disease. And then using the 3-minute thesis format, we got the students to present their grant proposal to the class. We then gave them face-to-face feedback and the opportunity to watch their grant proposal, okay? So what I'm gonna do now is I'm going to violate the rules of the Picture Future, and I'm actually going to show Sarah Ireland's presentation. It will go for 3 minutes, hence the violation, but she really illustrates how to use concepts that we all know. An example, she'll be using baking to describe a really complicated grant proposal. This is one of the most complicated grant proposals I have ever read, and she beautifully used baking. So we'll just wait for the 20 seconds to go, and then hopefully the sound will work and the mouse will work. Why is not the mouse working? Okay, here we go.
Hello, my name is Sarah, and I'm going to talk to you about my research grant proposal. One of the features of Alzheimer's disease is the deposition of amyloid plaques in the brain, which are essentially sticky clumps of a substance called amyloid beta peptide. This peptide is produced by the processing of APP, or amyloid precursor protein, which is damaging to the brain. Understanding how APP is regulated is important to further our understanding of the disease and to find better treatment options which target the mechanisms and not just the symptoms. To describe how proteins such as APP are made, we will use the analogy of home baking. So the initial idea for the cake comes from the professional baker's mind. This is where the initial information is stored, and this represents the DNA. In order for you to reproduce the cake, this information must be written down into a form of instructions that you can understand. This represents the transcription from DNA to messenger RNA, or mRNA, and the instructions represent the mRNA. You then take these instructions and convert them into a delicious cake. This represents translation, and the cake represents the protein. Research conducted in 2015 identified a G quadruplex near one end of the APP mRNA, i.e. in the instructions coding for an APP-flavoured cake. G quadruplexes form when a single strand of genetic material such as DNA spontaneously folds in on itself, and these structures can have important roles in determining whether proteins are made or not. Their research found that the G quadruplex had a negative effect on APP levels, such that when they disrupted its structure, there was an increase in APP. So imagine on the step telling the baker to add the bicarbonate of soda, the paper had folded in on itself, so you could no longer see to add it, and there was therefore less of the APP flavoured cake. Their research, however, did not explore how naturally occurring substances in the cell may affect these G quadruplexes. DHX36 is a protein known to bind and to unfold these structures. This proposed research aims to explore the role of DHX36 on APP levels by comparing the APP levels in cells where we've artificially decreased DHX36 production to control cells. So imagine in our analogy that your friend represents DHX36. She comes into the kitchen and she unfolds the paper so you now can see to add the bicarbonate of soda. We hypothesise that by decreasing DHX36 production, we will decrease the levels of the bad APP. And therefore, there will be less of this unfolding, less of this bicarbonate of soda added, and therefore less of this APP. This research could lead to novel treatments for Alzheimer's disease in the form of a DHX36 inhibitor. Hello, my name is Sarah, and I'm going to talk to you about my research. Hello, my name is Sarah, and I'm going to talk.
Right, so the student voice. 62% of the students found this a worthwhile experience, and 93% of students found it worthwhile seeing their talk via Panopto. So we do recommend that. Here's a couple of comments that fed back to us, and that is my 6 minutes and 20 seconds. Thank you for listening.